I'm here to introduce you guys to Felix. Who I'm sure many of you guys know. Well, this is our last journal club together, and so I would like to say a few things. First, Felix, we love you very much. Also, Google Translate was used extensively in the making of this movie. And lastly, this video is not really finished, but we don't think it will ever be.
Oh my god, it's so quiet.
No, there's a slider.
No,
Increase the volume.
Wait, hold on. Hold on. I think we need to.
Just done it.
Oh my god, yeah, let's get that in there. Alright.
I think. Can you pause? Wait, don't look.
I think it's working.
No,
Yeah, it's working.
Es heißt, in jedem Jahrhundert der Menschheitsgeschichte muss jemand kommen, der die schrecklichen Kosten für die Befreiung der Welt von Unwissenheit trägt. Einige können den Fluch nicht ertragen und werden von den schrecklichen Gewichten in den Wahnsinn getrieben. In den Eingeweiden der Erde verzweifelten große Männer darüber, wer als nächster das schreckliche Gewicht tragen könnte, ohne darunter zu zerbröckeln. Sicherlich können wir diese schwere Last nicht auf bloß ein Kind verfrachten, sagte Jclub. Verdammt, Nietzsche. Ich bin nicht mehr Mut als Sie, aber etwas muss mit Reagan geschehen. Außerdem sprachen die Prophezeiungen von einem solchen Schnurrbart. Aber die Überschlange. Kann es auch kontrolliert werden? Das Kind wird es als Fluch zu tragen und sie ganze Leben lang. Wenn es getan werden muss, werde ich es selbst tun. Im Schatten des Kalten Krieges, das Gespenst des Kommunismus, blieb ein neuer Staat ohne flexibele Ideologie überleben. Und so, mitten im Blut der Turbulenzen einer Welt, auseinander von Ronald Reagans Amerika, sollte Nietzsche den auserwählen. Als Felix und die rote Schlange entstanden, ersten das der Berliner Mauer. Eines neuen wahren Triumphes, der Erkenntnis, der ständigen Wahrheit überführt. Den Preis, den der Junge zahlen musste, ist akzeptabel in dieser Umständen. Als Kind war der Junge sanftmütig, gutmütig und freundlich. Von seinem Geburt an hatte er große Liebe erwartet. Er suchte Freundschaft über alles. Aber die Schlange, die sich wütend in seiner Tasche brütete, fuhren alle Kinder weg. Selbst die kleinsten seltenen Widersprüche. Der Wille der Wahrheit über ihr hässliches Nein, geh nicht, schrie der Junge. Obwohl der Junge sich immer über die anderen Kinder zu schädigen beschuldigt und fluchte dieses unerträgliche Qualen. Im Herzen spürte er auch etwas Großes im Verbund mit dem ewigen Freund in seiner Geschichte zu bearbeiten. Und von Zeit zu Zeit, so würden sie wach liegen und sich gegenseitig anfuchsen. Und so begann sein Dienst. Der Junge reiste über die Insel Zypern, um die wahre Herkunft der Überschlange erfahren zu bezeichnen, seine Lehre in Jclub und Reptilien zu suchen. Regeln begann, aber spürte, glaube ich, die Botschaft der Überschlange betont, alle seine Arbeit rückgängig zu machen. Kam der junge Mann von seinen transzendenten Visionen zu locken. Reagan bot ihm viele magische Geschenke, darunter das Buch der Furzprophezeiung, die Machtschleichen und Teile vorherzusehen und lebensgroße Poster von nackten und zügellosen jungen Männern. Doch die Überschlange blieb steif und widerstand den Versuchen. Als der Junge spürte, dass es Zeit war, sendete er die Überschlange ein Handelsschiff, um sie in New York Hafen zu schmuggeln. Auslebenden von Reagans obkursiven Regeln in seiner Fantasieforschungseinrichtung John die Wahrheitsziffer erstaunt über Schnurrbart, der vorausgesagt werden musste. Menschen flüstern über die Straßen über die Gerechtigkeit seiner Untersuchung und der Rundheit seiner Brille. Zweifels an des dogmatischen Kreativismus begannen in den Straßen zu vibrieren. Und alle waren sich einig darüber, wie unglaublich gut es sich anfühlte. Aber wie viele Geschichten endet diese in einem sofortigen Riechen einem eigenen Furz in der Hand. Eines Nachts Reagan und seine Anhänger hatten zu viel gesoffen, versammelten sie sich in ihrem Penissen und Mistgabeln, um den Jungen auf seinem Weg von New York nach Boston anzugreifen, wo er zwecks seiner Reise durch Nordamerika hinfuhr, um seinen Samen zu säen. Als Reagan schwang das Schwert des Straßenmagneten auf den Jungen, sprach sich die Überschlange in den graugrünen Klinge und stellte am Ende seinen Freien aus der Tasche, die in seinem ganzen Leben im Gefängnis war. Rief der Junge. Die Überschlange war sauber in 2 Hälften getrennt und lag keuchend zu seinem Knie. Der Junge nahm die Schlange in seine Arme und beschauderte überrascht den Atem der Strenge. Mein lieber Freund, Sie müssen weitermachen ohne mich. Ich weiß jetzt, dass die Überschlange aber wirklich euch alle auch. Der Wille der Wahrheit. In euch die Schlange und der Geist sind sein. Dein Wort ist Vertrauen in deinen Felix. Der Junge hob den Kopf und Ronald Reagan vor Wut in die Augen zu stopfen und rief: Ich bin der Übermensch. Um diesen Stein liegen nach Linien, Lücken und ihre Pfeile bedingen. Und wenn sich der Staub erzogen hatte, alles, was von der Übermenschen blieb, war ein Schnurrbart und eine kleine Brille.
Who wants to finish? For today. And with that, I give you Felix.
Hello?
Oh, sorry.
I made the mistake of listening to the narrator instead of reading, and that made no sense at all. Second, I guess it's very dangerous to have special interests like snakes, because they make for a good topic. And third, I really like the Bavarian shorts. That was thank you for the kind introduction. I think both of the introductions today are very fitting of our class's goodbye session today. So given that this is the last presentation of our class, I got a little bit sentimental, and I remembered the first time our class gave the journal club, and I went back and tried to come up with the poster for that first session. And it turned out it was our very own Julie who gave that journal club shortly after in October 2013, shortly after we were all rocking campus, and still very shy, still very naive. And I think her title is great and really encapsulates how most of us were probably feeling at the time. OR, phone home, how olfactory neurons lost in space find their way to the olfactory bulb. But I think also that I speak for everyone when I say that while these Friday afternoons have been great, we all are fine, not sad, to move on and hand over the torch to the next generation. So I'm going to talk today about gene regulation in a developmental context. And I think you all know that turning genes on and off during development determines the identity of cell types, of tissues, of organs, and ultimately of our body plan. And some of the genes that are perhaps most famous for regulating the environment are Hox genes. Hox genes are transcription factors that are known for patterning the vertebrate body plan, and they exist in 4 families of paralogous genes that amazingly are linearly arranged on the chromosome in the genome, and in that same sequence are collinearly expressed the body axis from anterior to posterior, where they then, among other things, specify the identity of vertebra. So in this context, it's easy to see that if you disrupt this expression, or you get an abnormal expression pattern of Hox genes, you can observe something like transformations of vertebra, by which I mean that a vertebra assumes the identity of the vertebra that lies before, anterior, or after, posterior to itself. So the story that I want to tell you today begins with such a transformation. And those same authors whose paper I'm presenting today investigated a mouse mutant by the name TS, which stands for tail short. And this mouse mutant was known since the 1950s, but no clear mutation had been identified that would be underlying its phenotype. And so the phenotype, as the name says, tail short, is a short tail. There's some other changes. Those mice have severe skeletal abnormalities. And one of the striking features of this mutant strain is that they possess a 14th rib, strongly suggesting that the first lumbar vertebra that usually doesn't possess a rib basically was interiorly transformed into a thoracic vertebra and now possesses a rib, thus adding up to 14 ribs. With basically what I told you about Hox genes right now, to uncover the mechanistic problems behind this phenotype, what the authors first wanted to do is see maybe if there's a change in Hox gene expression or expression boundaries, right? That is the most, the next conclusion you would draw. And surprisingly, they found that there's no difference at all in Hox gene expression levels or Hox gene expression boundaries. So in this situation, what they next wanted to do is to positionally clone this gene and see, well, what's really going on, which they did, and they identified a deletion in the ribosomal protein RPL38 as the underlying causative mutation, which is striking, right? You wouldn't expect a ribosomal protein to be somehow involved in patterning problems of the vertebrate body, if at all there is a problem with translation, then you would expect multiple phenotypes, since translation is so globally important for the organism. And consistent with these doubts, when they assayed global protein levels in those mutant mice with the reporter construct, they basically found that there's no difference in global translation activity. So again, something that doesn't really fit in the picture, what's going on. Now you have to know that these assays basically will not pick up minuscule differences. If you have differences in translation between just a few genes, then those global assays might not be sensitive enough to pick up those differences. And so what they did is they went back to their Hox genes and directly assayed the concentration of those Hox genes at the protein level, and this time landed a hit. There was a decreased Hox gene protein level for just a subset of those genes in the mutant mice. Okay. Now, what might be causing this, these different levels? So for that to find out, they basically looked at the mRNA abundance in fraction. They fractionated somite tissue and looked in fractions that contain polysomes, which are ribosomes that are attached to an mRNA transcript, suggesting that they are in the process of translation. And when you look at those polysome fractions, you see that for those Hox genes, and here they are listed out, I want you to focus on Hox A4, A5, A9, and 11. You see that they are severely or significantly less associated with those polysomes than any of the other Hox genes or any other genes. So from this and from some more experiments that I don't have the time to go over today, they concluded that somehow RPL38 controls the ADS complex formation, so the formation of the ADS ribosome on select Hox mRNAs. Now this is only one half of the explanation, right? And what my paper is basically trying to address is trying to look at the other side. How is this regulation possible? What is so specific about those Hox genes that they can be regulated at the translation level? My paper is entitled RNA Regulons and Hox 5' UTRs confer ribosome specificity to gene regulation. It is coming out of the Baier lab at Stanford University. It was published in Nature on the first day of this year. As I just told you, they knew that the genes depicted by these boxes here are translationally regulated by RPL38, but they didn't know how this happened. And so when we look at what typically happens in eukaryotic translation, then I think it's pretty clear to most of you that there is this cap, this methylated guanine base at the 5' end of the transcript, that recruits the 60S and 40S subunits of the ribosome, which then initiate translation at the neighboring start codon. We call this process cap-dependent translation, and it is thought to account for most translation events in eukaryotic cells. Now, if you look at this, you could imagine how something weird in the mutants might be going on between the 3' UTR and RPL38, or the 5' UTR and RPL38, or maybe even the cap itself. And to decide among these options, you would really need to take a systematic approach. But instead, what the authors did is they made two assumptions. Assumption number one, they assumed there's something in the 5' UTR that interacts with RPL38. And assumption number two, we want to be able to distinguish between cap-mediated translation and any other form of translation initiation from within those UTRs. And so they designed an experiment to address these questions, which consists of this reporter construct, this biosystronic reporter construct, in which you clone your 5' UTR question directly before a firefly luciferase coding sequence. You then have a second cestron in here, which is the gene for vanilla luciferase, with just another luciferase that has green bioluminescence, while the firefly luciferase has red bioluminescence. And you know that this transcript is going to be capped in the process of transcription, so anything that you see under that is green suggests that you have cap-mediated translation. Anything that you see that is red, it suggests that you have something in here that initiates translation of this firefly luciferase. They made sure that there is no ribosome slippage, and you could see how the ribosome slides through and thereby initiates translation. So when you do this for all those genes, you see that again those 4 genes, A4, A5, A9, A11, have a greatly increased translation initiation activity, which is not true for all other genes except for A3, HOX-A3. Now, consistent with the transcript specificity of RPF38, if you now knock down RPF38, you see that while those 4 genes are affected by this knockdown, they have reduced translation initiation activity. HOX-A3 is not affected. So that basically confirms the earlier paper. Now, what exactly is it in those 5' UTRs that is able to initiate translation? To answer this question, they conducted a number of deletion experiments in which essentially you remove bits of the 5' UTR and then look at how this affects this activity. And strikingly, you see that if you remove just the last 300 base pairs, then you see that this construct, the first 944 base pairs of this 5' UTR, has very little activity. But then if you take this last bit only, you see there's significantly increased translation activity that truly, or admittedly, doesn't recapitulate the full level, but it's the translation activity of the full construct, but it truly gets to a significant level. And from this, they concluded that this region here is basically a minimal segment that is capable of inducing translation. Those same segments exist also in the other genes, but they vary greatly in size and also in position within the 5' UTR. Now, this is all interesting, but we still don't know what this segment really is and does, right? And so what the authors did is they basically had a look at its structure and found it to be, surprisingly, found it to be a 3-dimensional RNA structure that has asymmetric bulges, pseudoknots. There's a lot going on. Those structures are also functionally irrelevant. If you mutate, for example, this loop here or this loop here, or both together, you see that you get a decrease in translation initiation, suggesting that they are essential for this function. At this point, they went in the literature and looked up what those structures might be, and they found a similar structure that was known from viral mRNA, and is called an internal ribosome entry site iris, which in viral mRNAs such as hepatitis C virus, they sit in the 5' UTR, and they are able to recruit the chromosome, the ribosome by themselves, independent of cap-dependent translation, which is very useful to them because in situations of cellular stress, the cell basically shuts down all translation, and the virus, which is invading the cell and has an interest to be replicated and translated, basically is thereby able to keep that business up and running. So based on this similarity, from now on they call these minimal segments iris segments, and they call the translation mode iris-mediated translation. Turns out that these iris segments are evolutionarily conserved. Here you see the HOX-A9 5' UTR of a number of vertebrates, whereby you see that most of the 5' UTR is actually not conserved, but this minimal iris segment is very conserved across all those species. It's also functionally conserved. If you take the zebrafish version of this iris segment, you implant it into murine cells, you see as much activity as with the mouse's own iris segment. Now, this all strongly suggests that this iris segment is able to recruit the ribosome, but obviously we want to see that backed up with evidence. That's what they did next. They basically tagged these UTR and or iris segments with biotin and exposed them to a cell lysate, which then basically can pull down proteins in the cell lysate, and you can quantify this by Western blot analysis. And what you see is that both the full-length 5' UTR and the iris minimal segment, but not the UTR without the segment, are able to pull down proteins both from the 40S and from the 60S ribosomal subunit. And that's also true for ribosomal RNAs, 18S and 28S RNA. So now, I think we're pretty close to understanding what this iris segment is doing, except we haven't looked at what it's actually doing in the process of normal translation. We've always done this in a bicestronic reporter. We haven't looked at this situation. What happens if you take the iris, the 5' UTR with the iris segment, you just put it upstream of a firefly luciferase, whereby you would expect that this transcript is capped during transcription. And here they really landed a surprise that nobody expected, because they noticed that if you do this, if you remove basically this iris segment from this construct, you don't get any translation activity, despite the fact that this transcript, right, should be capped. So you should see translation activity, but they didn't, and that was very striking. Something else must be going on here. They went back and did more, deletion experiments. And found that if you only take these first 300 base pairs and you put them in this system, you see almost no activity, suggesting that these 300 base pairs are the ones that are inhibiting CAP-mediated translation, and that's why they call this segment the translation inhibition element abbreviated by TIE. Turns out that also the other HOX genes have the same segment, this TIE segment, but it varies in size, it varies in position, it's not conserved. And then as a second proof, so to speak, they pasted this TIE segment upstream of a beta-globin 5' UTR, which is usually able to induce very high translation, but if you have the TIE segment, then, so they pasted this TIE segment upstream of the beta-globin 5' UTR, and then observed that if you have this TIE segment present, there's no translation activity compared to just the 5' UTR by itself. Now, as a final step, I'm sure you already guessed that what they wanted to do next, take this into the living system, and for this purpose, they produced a knockout of the iris minimal segment. So you remove those 300 base pairs. And then if you look at the mice, strikingly they have an interesting phenotype. They only have 12 vertebrae, and the heterozygotes, they have, and 50% of the time they have also 12 vertebrae, but you see that there's sort of a little bit of a vertebra growing out there. If you look at the protein level, you see that HOXA9 is reduced in those somites, so that's fitting in the picture. Global mRNA levels of HOXA9 are not affected. That's also fitting in the picture. And if you do that fractionation analysis again, you look at how much of the HOXA9 mRNA is associated with the proteasome fraction, then you see that it's greatly reduced in the iris knockout, but then if you look at the frequency in the pre-polysomal fraction, you see that it's greatly increased. So there's a shift of where those mRNAs are located. So I think from this we can arrive at this take-home message. This paper describes a new mode of translation. They call this iris-mediated translation. It's relevant for translating Hox genes, which are critical for vertebrate development. It works by these iris segments interacting with RPL38 to trigger translation, while the Thai inhibits cap-dependent translation. Okay, so what do I think about this paper? I think it is an interesting paper that presents a new mechanism of translation. What I was sort of missing throughout this paper was a rationale. Like, why would you do this? Right? Like, why does the cell need this alternate mode of translation? How many other genes are affected? I didn't tell you so far that there is precedence. There is some genes are known, stress response genes in eukaryotes that have iris segments and that can do the same thing that basically the virus is doing with its own genes. They can recruit the ribosome and in situations of stress when cap-mediated translation is shut down, and thereby uphold their own translation and try to rescue the cell from the stress. So there at least there's a clear rationale, right? But what's the rationale in the case of development? Like, why would you need to uphold the development even in situations of stress? And I think that is sort of not answered well. What the authors suggest is that the iris is very conserved. It's known from insects. The Thai is not very conserved. It evolved after basically, so amphibians and fish don't have it, so it evolved after this split. And so they paint this picture where because those amphibians and fish, they basically lay their eggs in very harsh environments, and it's beneficial to have the system that continues to translate your proteins to withstand that stress. And then after those fish, basically the Thai was added as a segment that can regulate this earlier system. I don't know if that's convincing. I have my doubts. So then, second point is, I asked myself the question: how can RPF38 interact with those 4 Hox genes if their iris segments and the Thai segments are really not very similar to each other across those Hox genes? Especially the Thai segments are not very well conserved. Is it just the secondary structure that is giving the specificity for this interaction? Is there some other proteins involved that interact with RPL and with iris segments, so there's no direct contact? And lastly, I wanted to draw to your attention that while a knockout of RPF38 and a knockout of HOXA9 get the same phenotype of 14 ribs, if you knock out the iris segment, you get 12 ribs. But we're really talking about the same interaction here, right? Like, it's iris interacting with RPF38, so I think it's fair to assume that you would get the same phenotype, but you don't, and they don't comment on that. So with that, I would like to thank my coaches Victoria and Vlad, and I'm happy to take your questions.
So, I know that viruses have irises and that we use them in cloning all the time. Is there any evolutionary relatedness between animal irises and viral irises?
So they both have the same sort of secondary structure elements. Did I not mention that? I meant to say that. So they both have those bulges in conserved UDCs and pseudonuts. So there's some, you can build the case that those secondary structures may basically mediate the interaction.
Do we know which one came first, if one came first?
You mean the virus or the Good question. I would say that the virus, but I'm not sure.
Did they look at all that translation efficiency coming off of these iris transcripts, right? Because that could, we're trying to like come up with a way of why would you want this, and if there's some sort of tuning of the efficiency off of the
They did. A number of controls that they could have done, like for example, an RPL38 knockout in the pull-down experiment to see if you can still, if those iris segments still pull down other proteins. They actually haven't done those controls, and this is one of the, they looked at the mRNA levels, but the transcription.
The TS mouse, they originally, they have any other phenotypes?
Yeah, so they have a short tail, right? They have 14 reps, and then they have like a number of skeletal abnormalities that affects a fraction of those mutant mice. They don't comment on behavior or anything like that, no.